Hey, CW Bio, Mr. Kennedy here, and this is part 2 of your Chapter 11 lecture notes, where we get to, meet a guy by the name of Gregor Mendel. Gregor Mendel, he was a buddy of mine. He was alive in the mid-19th century, an Austrian monk who carried out some important studies in the world of genetics or heredity. Basically, he's the first guy to explain how characteristics are passed from parents to offspring. Prior to Mendel, everybody thought that kids, any future generation that was produced was basically just kind of a blended milkshake of whatever the parents were. So it's kind of like saying if your mom had black hair and your dad had blonde hair, that you were just destined to have brown hair and there was no in between. Okay. We now know that that's totally not true, thanks to Gregor Mendel. Gregor Mendel studied these things that we see in the mirror, and, in kind of a blanket term, we call all the stuff we see in the mirror traits. And as you just learned from my video on why meiosis, those traits are determined by the genes that we inherit from our parents, right? Mendel was the first person to successfully not only figure this out, but to predict how those genes would appear and in what mathematical proportions in future generations without having to actually make future generations. His work, the complete explanation of his work, is now the science of genetics, right? The branch of biology that studies heredity. Let's look at how he did it. So the first thing we're going to dive into is his test subject, if you will. Mendel chose to use a common garden pea in his experiments. He probably had lots of reasons for picking the common garden pea, not the least of which was, as a monk, there were no grocery stores. So if you wanted, vegetables for dinner, you had to grow them. And so these were growing all in the garden that he was responsible for tending. And as he walked around the garden, he probably noticed some things about these garden peas that were a little odd, especially in light of current beliefs at the time. For example, in these garden peas, they reproduced sexually, which means they produce male and female sex cells that we've learned are called gametes. And as they reproduce sexually, the male gamete formed pollen, right? And that pollen was formed in the male reproductive organ of the flower, and the female, it produced the female part of the flower produced an egg. And when you bring those two together, we have fertilization. That fertilized egg then becomes called a zygote. This is new stuff. You got a flower that has male parts and female parts to it. Male parts that only produce the male pollen, and then a female part that only produces an egg. No one knew this in the mid-19th century. I know, it's crazy because everybody nowadays takes it for granted. But in the mid-19th century, this is cutting edge, okay? Anyways, so when the pollen meets the egg, that's fertilization. We get a zygote, okay? The transfer of pollen grains from the male reproductive organ to the female reproductive organ, that's just pollinization. But when the actual embryos, not embryos, when the actual nuclei of these reproductive cells come together, that's fertilization. Now, Mendel probably also noticed some other weird things that these garden peas had going for them. There were no halfway in between traits. You know what I said at the beginning where people thought that, cats, dogs, fish, frogs, even humans were just milkshakes of their parents and some sort of blend between the two? Well, when Mendel's walking around the garden, he sees plants that have this purple color to it or red color. And then there's white ones, but there's no pink. What's up with that? There's tall plants and then there's short plants, but there's no medium-sized plants. I know what you're saying. In your head, you're like, Mr. Kennedy, it's just a pea plant. Who cares? Well, okay, there's nothing on television in the mid-19th century. So what's Mendel got to do besides walk around the garden and go, dude, where's all the medium pink colored plants? Right? So he decides to start doing some experimentation on these guys to see what is going on. So he basically goes through these plants and he wants to control their breeding So he goes through flower by flower and removes the male part of every one of these flowers, like literally cuts the little, anthers out. So now these flowers can't produce pollen. Then he collects those and determines whom or what he wants to breed or crossbreed, and takes a little paintbrush and gets pollen from a specific plant of his own choosing and puts that on the female part of a specific plant of his own choosing, and then he covers the flower up. Now, I don't know if you've ever seen a pea plant before, but these guys have lots and lots and lots and lots of flowers. I don't know about you, but I don't know if I've got the patience to literally brush pollen on 100 different flowers and then cover the thing up, day in and day out. Mendel did, right? So to his credit, that's what he did. He hand pollinated these things, controlled who bred with whom. So when you cross two different plants, this is what we call cross pollination, right? And through this cross pollination, Mendel could basically create pure breeding strains of plants that were interesting to him. He controlled who the parents were in his crosses. So he created populations of plants that were pure for 7 different traits, like red flowering plants that always produced red flowering plant babies, and tall plants that always produced tall plant babies. And you get the idea, right? Green pea plants that only produced green peas, and yellow pea plants that only produced yellow peas, and axial plants that only produced axial flowers. There were 7 of them. Just take my word for it, okay? So then Mendel decides, okay, well, I want to see what happens if I cross a tall plant with a short plant. Am I going to get medium plants? What? Let's see. So he studied one trait at a time to control all the variables that he could in his data mathematically. The tall pea plants that he worked with had been from populations of plants that were tall for many generations and only and always produced tall offspring. So here's what he did. He took short plants that were of the same stock as the tall plants, and he crosses these two. Okay. He wants to produce what's called a hybrid. A hybrid would basically be a cross between a tall plant and a short plant to see what you're going to get. Okay. Mendel's first experiment is called a monohybrid cross for this reason. He crossed 2 parents who were different from each other in only one way, right? And that one way was their height. Everything else about them was the same. Their flower color was the same. Their pea color was the same. Their flower position was the same. The pod shape was the same. Everything was the same except one was tall and one was short. So when he does this cross, okay, it kind of goes without saying, if you cross a tall plant with a short plant and you go by the current belief system of the time, all the offspring had better be medium sized, right? So Mendel selected a 6-foot tall pea plant that came from a population of pea plants that were all 6 feet tall. He cross pollinated that tall plant with a short plant, and lo and behold, all the offspring grew as tall as the taller plant, 6 feet tall. What's up with that? Mendel expected medium-sized plants. Now, he could have thrown the towel in right here and said, you know what, I quit, whatever, it's just pea plants, who cares? Let's go make some soup, all right? But instead, he decided to go one step further, okay? And that one step further is that he took those offspring from that first cross and he allowed them to self-pollinate. Now, this is something that you can do with plants, but you can't do this with animals because, it's just not possible. Anyways, so he allowed these tall plants to self-pollinate. So that's basically a tall plant is crossed with a tall plant. So if I cross a tall plant with a tall plant, what do you expect the outcome to be? Yeah, tall plants. Well, dude, look what he got. After the seeds formed, he planted them. He counted more than 1,000 plants in the second generation. 3/4 of the plants were as tall as the tall plants in the parent first generation, but 1/4 were short. What's up with that? Where did those come from? Because, hey, I just crossed 2 tall plants. How come their offspring are short? Weird, okay? 1/4 of the offspring were short as the short plants in the parent generation. In the second generation, the tall and short plants occurred in a ratio of about 3 tall plants to every 1 short plant, okay? Which is bizarre, Because, again, where's the medium plants? And, dude, where did these short plants come from in the second generation? So the original parents, they were true breeding. Mendel called them the P1 generation. The offspring of the parent plants are known as the F1 generation. When you cross two F1 plants with each other, their offspring are the F2 generation. Keep all this rattling around in your brain because you'll need it as we keep talking about what happened here. So here's the different traits that Mendel crossed. He had 7 traits that he studied: height and flower color and pea color and pod shape and pod color. He did the exact same cross I just described for you for all 7 of these traits. And guess what? In every case, he found that one trait of a pair seemed to disappear in the F1 generation, only to reappear unchanged in 1/4 of the F2 plants. Weird. And there was never a medium like in between. There was never like a greenish yellow seed. It was either green or yellow, like nothing in between. No medium plants. So this led Mendel to conclude that each organism basically has 2 factors that control each of its traits. One from mommy, one from daddy. We now know that these factors are called genes, and they're located on our chromosomes. Genes exist in alternate forms that we, refer to as alleles. You learned that in the last video that I just gave you. And from what Mendel's first studies taught him, he concluded that an organism's 2 alleles are located on different copies of a chromosome, one inherited from a female parent, one from the male parent, and that one could actually dominate or hide another. Mendel called the observed trait in the F1 generation the dominant trait, and the trait that disappeared only to reappear in the F2, the recessive trait. Mendel concluded that the allele for tallness in plants was dominant to the allele for shortness, along with so many other things, like red flowers are dominant to white flowers, and yellow peas are dominant to green peas. I know that's weird sounding, but it's true, okay? When he recorded the results of his cross, it's customary to use like the same letter for different alleles of the same trait. So when we're talking about a tall plant cross with a short plant, you're going to use the first letter of the dominant trait to designate that allele when you do your crosses. So you look closely here, you can see that I've got a parent here that is big T, being crossed with a parent here that is little t, okay? Each parent is going to produce a sex cell, a gamete, which is what we see going on here, and contribute that to the next generation. And so the offspring each inherit one allele from each parent. So now the offspring is big T and little t, thusly, okay? Being big T, little t, that's what we call being heterozygous. As long as you have a big T, that's what is expressed. That's what we see, because the big T is dominant to the little t. Okay, so the uppercase letter is used for the dominant allele, lowercase letter used for the recessive allele, but it's always the same letter, like it's a T, okay? We don't use big T for tall and then S for short, like that doesn't fly, okay? Second thing for you to consider is this. Mendel came up with what we now call the law of segregation. The law of segregation states that every individual has 2 alleles for each gene, and when gametes are produced, each gamete receives one of those alleles. So that means during fertilization, the gametes randomly pair to produce 4 combinations of alleles. That's genetic variation, people. That's awesome, okay? So the law of segregation basically guarantees that you can't put from a single parent both alleles for a specific trait in the same sex cell, because when you combine that with a different parent, like there'll be all kinds of confusion. Like you would end up with too many chromosomes, and who knows what the offspring would look like. So this is how that works, all right? So if you have a parent that is big T, little t, right, they're going to produce 2 different kinds of sex cells, a big T sex cell and a little t sex cell. Same thing over here, okay? And then the combinations of those sex cells will determine what the offspring look like. You could get an offspring that's big T, you can get offspring that are big T, and you can get offspring that are little t, okay? And it's just whichever sex cell gets there first, so to speak, that determines what the offspring will eventually look like. Keep in mind that as long as there's a big T, right, then in the case of these pea plants, the offspring are going to be tall. Okay? If it's little t, then they're going to be short. So what we're seeing outwardly and what we inherit actually has names. Okay? So the way an organism looks and behaves is called its phenotype. The genetic combination or allele combination that it inherits, that's its genotype. Knowing an organism's genotype, well, can't always like guarantee that we're able to nail the phenotype or vice versa, but it goes a long way into making it possible. Okay, so if you have two of the exact same letters, big T, then you're going to be what's called homozygous. Homozygous. If you have big T, true breeding for tall, right, you're guaranteed to be tall. Both alleles are the same. You could also be homozygous recessive, which means that you have two little t's. If you have one big T, one little t, then you're heterozygous, okay? And in the case of heterozygousness, I don't know if that's a word, but it sounds cool. Remember, the capital letter, the dominant one, is the one that we are going to see. The recessive one is going to be hidden by the dominant one. Okay, so in the fact that this case, this plant has one dominant allele for tallness and one recessive allele for shortness, well, that plant's going to be tall. Okay, now Mendel did a variety of other crosses, and we're going to spend lots of time in class like practicing monohybrid crosses and getting really comfortable with those. But he also did one other cross to help kind of satisfy Mendel's laws that I'm just going to share with you here in your notes, and it's called a dihybrid cross. In a dihybrid cross, we're going to cross two characteristics, two traits from two different individuals at the exact same time. Okay, so it looks like this. Mendel took a true breeding plant that had round yellow seeds and crossed them with a true breeding plant that had wrinkled green seeds. Look at all those alleles. Big R, big Y, crossed with little r, little y, little y. Whew! He already knew that round seeded trait was dominant to wrinkle seeded trait. He also knew that yellow was dominant to green. So when we put these two things together, we just kind of want to see what happened, right? So in a dihybrid cross, looking at what Mendel did here, again, there's no blending of traits. The important thing that Mendel discovered was, as you look at this, there's also the possibility of new combinations of traits that didn't exist in the parents, okay? These new combinations of traits go back to what the parents originally looked like, a round yellow seed and a green wrinkled seed. So if you cross those two parents, you would assume that all the offspring would look like one parent or the other. Well, look what he found out. Like, you could get a round green seed and a wrinkled yellow seed. Neither parent looks anything like that, okay? So Mendel let the F1 plants pollinate themselves. He found that some of the plants that produced round yellow seeds and others produced wrinkled green seeds. He also found some plants with round green seeds and others with wrinkled yellow seeds, which was weird because none of the parents had that. So he found a mathematical formula to kind of represent what he saw, a ratio of phenotypes of about 9 to 3 to 3 to 1. 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Okay? Because we got new combinations of traits that didn't exist in either parent, Mendel came up with his second law of genetics, and that second law of genetics is called the law of independent assortment, which basically means you can inherit traits independently of one another. It's kind of like saying, in these pea plants, there's no guarantee you're going to end up with a round seed or a wrinkled seed, a yellow seed or a green seed, because they're not connected to each other. In humans, if you just want kind of like a lame example for people, like if your mom had blonde hair and your dad had brown hair, like in your family, there's no guarantee that every female in your family has to have blonde hair because it's not connected. You know what I'm saying? Like you could have guys in your family with blonde hair and girls in your family with brown hair. Like they're not connected. Okay, that's the law of independent assortment. Now later, Mendel, he proposed all this stuff. He shared it with the scientific community, and most people looked at him and said, huh? Like, I don't know what you're talking about. And the reason they didn't know what he was talking about is because he dropped one of these guys on him. In 1905, a man by the name of Reginald Punnett, who was an English biologist, devised a shorthand kind of like multiplication table for predicting what offspring would look like generation after generation. And we're going to get lots of practice with these things, but the general world, like, they weren't educated in statistics. They didn't know what Mendel was talking about, and at the end of the day, his work largely went unappreciated for a number of years. Anywho, Punnett squares are a cross multiplication table that will allow you to predict the possible genotypes and phenotypes of offspring thusly. A Punnett square basically takes the alleles from each parent and puts them on the side and the top, like you see going on here. And then you add an allele from each parent to each box. Okay? So as you do that, you fill in these boxes, and you will eventually come up with a ratio that we will practice in class that will predict what these offspring look like. Okay? It doesn't matter which set of gametes are on the top or which are on the side. Each box is filled with the gametes from above and to the left of the side of the box. And then you can see that each box contains 2 alleles, one for each possible genotype. And after those genotypes have been determined, you can determine the phenotypes and come up with ratios for those and report those out, like the 3-to-1 ratio or the 9-to-3 to 3-to-1 ratio. Don't worry, we'll do lots of practice problems in class. Now, the dihybrid cross is muy grande, right? It's way bigger. But the idea is still the same. We're taking alleles from the parents and we're putting them on the left and across the top, and we're filling the boxes in as you see here. Okay, it's just that now we have 16 boxes. Okay, and again, don't worry, I'll practice this with you in class. The key is you look at this. If we have 2 parents that are heterozygous, big R, big Y, little y, and you look at the offspring here, right, we get new combinations of traits that didn't show up in those parents, which is what independent assortment is all about. All right, ladies and gents, that, in a very quick, very simple nutshell, is how Mendel came up with his laws and what a monohybrid and dihybrid cross taught us. So we're going to practice this in class. I hope you've learned lots about Gregor Mendel thus far, and I'll see you next time.